Okay. Thank you, everyone, for coming to another one of our webinars from the NIDA Center of Excellence in Omics Systems Genetics and the Addictome. Today, we're going to hear from Elissa Chesler from the Jackson Laboratory, along with Erich Baker from Baylor University. And they're going to give us a great intro on to GeneWeaver and a little bit of a tutorial. We will put their slides up later at our GitHub site and send out a link. We will also post this entire presentation on YouTube. As we go through, we ask that because we're a large group that you keep yourself on mute. And if you have questions, go ahead and send those to the chat. And what we'll do is Rob has graciously offered to monitor the chat. So he'll be watching for those questions and we'll be able to break into the presentation at the appropriate times. And as usual, we'll also put the chat then also up on the GitHub page once we're done. Thank you so much, Elissa and Erich, for coming to show us. Show us what's going on with GeneWeaver. I will go ahead and turn it over to you guys.
Thank you, Laura. Thanks for inviting us. And I do have to thank the Rat Center for GWAS for the caffeine I received yesterday. I want to share with you all about GeneWeaver. We'll provide an overview of GeneWeaver and how it's set up conceptually, how it's organized, what some of its applications are. I'll talk about some of the emerging tools that we've been releasing and developing, and I will also provide information on how you can do a demonstration exercise on your own using the website so that you can become familiar with all of its functionality as you go. GeneWeaver's been in continuous development since approximately 2007. It's been through many iterations, and we've very recently, within the last couple of years, re-envisioned GeneWeaver as a suite of APIs. So it's quite modular, it's quite modern, and has a lot of new data in it, and we've been expanding the data model to incorporate now variants and other entities. So I'm going to kind of step back and say, what was GeneWeaver designed to do? And the driving problem that we're trying to solve is the integration of functional genomics data. We have in many different experiments in many different species now have the technology to characterize tens of thousands of genes and their roles in various very specifically defined traits and processes. And each of these processes are defined either by data curators or by people doing experiments who represent very sophisticated biological concepts into their research. So in the context of addiction, we might ask about gene expression as it's altered through many different processes of initiation of drug use, of extinction of drug use, reinstating drug use, and drug use in response to different environments, different genetic backgrounds, et cetera. All of these experiments are very exquisite, and they allow us to tie genes and genetic variants to very specific biological processes, each of which results in a list of genes. We have lots of different high throughput technologies now: genomic, proteomic, transcriptomic, genetic, but lots of others that emerge that allow us to en masse implicate lots of genes with diseases. And this includes high throughput screens of model organisms. So if we look at projects like the International Mouse Phenotyping Consortium, 8,000 genes are being tested on a large number of different physiological, anatomical, and behavioral assays. So all of those genes then get annotated to the traits that were measured. And then we have all the data that are collected in individual laboratories, written up in publications, and again, also used to annotate genes to terminology in the literature. So we have diverse functional genomic data, a lot of it collected in a high throughput context, and we want to be able to integrate this across many different organisms. And I know represented among the participants in this call are people who work with humans, mice, rats, potentially Drosophila, zebrafish. We're all asking very related questions. And one of the fundamental questions that we want to answer is, how does my research study relate to your study? Are we finding the same genes? And what is conserved and not conserved when we look across the different species? Can we compare different traits across species and ask which traits are relevant to the study of different facets of human disease? And GeneWeaver provides us a platform for sharing data and analyzing it to address these types of questions. So this is a slide from an older paper now in nucleic acids research, but it lays out sort of conceptually how GeneWeaver is built. We have a large data repository that contains functional genomics data represented as gene sets, and it's pulled from a variety of sources. Some published, some from other databases, and some contributed by users and curators. Using homology and gene orthology, we're able to map the identifiers used to represent genes and gene products onto one another and allow us to simply integrate that data using a bipartite graph structure. And so this is this bipartite structure shown coming in and out of the database. Basically, a query of the GeneWeaver database retrieves a set of gene sets and, in prototypical development, sets of gene networks, and aggregates them into a single data structure that can be interrogated to address multiple different problems using a suite of tools that allows users to apply different set operations very flexibly to the aggregate set of data. And so you may have a question that says, well, how do I refine? I have many overlapping QTLs from rat studies of addiction-related behaviors and physiological response to drugs and behavioral response to drugs, and maybe even some non-addiction related behaviors. How do these QTLs relate to one another? And if I look at their overlap, can I find one gene or variant that's associated with all of them or a subset of them? So we could take lots of data. We might also ask, are differentially expressed genes or genes regulated by cis-QTLs or genes for which there's evidence in other species, are any of the genes within my QTL interval, my genetic interval, also supported by functional genomics data that's completely independent of genetics? And this is very helpful when you map a trait in a species or a population for which you have large LD blocks. Any genetic analysis is going to have trouble resolving that locus, but all of these orthogonal experiments, these functional experiments, allow you to refine that locus. There's a lot of other applications, and one of the most unique to GeneWeaver is our hierarchical set similarity, which is represented in tile 7 in this panel. We basically enumerate all the intersections of all the gene sets in a user selection and represent them hierarchically. So you could say I've got 50 studies of anxiety-related traits. What genes are shared among which subsets of those traits? And this is done quite seamlessly from data derived from multiple species and multiple experimental platforms. So again, the way we bind that data is through gene homology and orthology. So we're looking across different species, and we're looking at all of the representations of that gene or gene product in those species, and using that as an identifier to bring together the data in that bipartite structure. And that ties together a wealth of diverse data. In our more recent development and currently not publicly available is that we're using now gene regulatory variants and gene regulatory features to start looking at the comparison of non-coding variation and regulatory variation across species. So we know the targets of the regulatory variants in many cases, either through expression QTLs or 3D genomics or other technologies. We know where regulatory features are, and we know that we can at least estimate the orthology of the target genes across species. And now we can ask which non-coding variants are predicted to have the same effects on those targets And so we can actually start aligning functional genomic studies in one species to variant mapping studies in another species. So you might say, well, are there mouse genes or mouse variants affected by the variants I've identified in a human GWAS? And when you pull those data, I'm asking that like I'm asking a question about a single variant, but you can actually do this comparison globally. So you could take a GWAS result, a vector of variant association scores, many of which are in the database now from LD Hub, and you can actually retrieve model organism experiments or model organism traits that are associated with the same collection of variants. So that's the type of thing that's enabled by the new data model. This is a figure from a recent review by Timothy Reynolds and a number of people actually I think on the call, including, well, a number of people are on the review. But we lay out this model and I refer you to it to get the details. But the basic idea is we have mouse and human genes, and we can, of course, extend this to the other supported species in GeneWeaver. These are regulatory variants, variants within genes, EQTLs, and the mouse genes themselves. And so these regulators regulate the gene. The gene has an ortholog in the human, and we could go in and ask questions about regulatory variants in a human. And so you can start constructing these graphs from mapping of variants in one species to regulatory features in another species and so on. So this allows us, again, more capability in binding together highly heterogeneous data for interrogation and analysis by scalable algorithms. That sounds complicated, but GeneWeaver.org is a browser-based tool set that allows you to easily access this functionality through a graphical interface and to work together collaboratively through its different sharing capabilities. So I'm going to go through some of the features of the website. I don't want to do a live demo because things always take longer than you think they will when you do, but we do have the ability to switch over to the website if people want to look at some things. So this is the homepage. On it, you see basic functionality. Any term that you enter into this search box could be, and it could be a combination of terms, allows you to interrogate the database to find gene sets related to traits, biological functions, drugs of interest, but also genes of interest. So you can retrieve any gene set containing your favorite genes. This tends to work best. This will give you any gene set containing at least one of the, or rather, well, depending on how you set up the search, one or more of the genes that you enter into it. There are other tools for looking for a list of genes and genes that highly represent the members of your list. Overall, we have tools for managing gene sets, so uploading a set of genes, organizing your sets of genes, a curation interface that allows you to set up a standing database query, a PubMed query, integrate those data into the curation interface, set up new gene sets, assign them to users to curate so you can work in a team among your lab group to bring those data into GeneWeaver, and then tools for analysis of gene sets that you've selected using the search page. Also under here are tools for looking at past results and rerunning past queries on either the original or updated versions of gene sets that have been uploaded. As I mentioned previously, there's a large number of types of functional genomic sets, either from genetic mapping or from a variety of gene expression genetics analyses and differential expression analyses and lots of other experimental techniques. So we've embedded a lot of other existing resources into GeneWeaver. Those include curated repositories, large experimental resources like Allen Brain Atlas, and many others. So again, lots of different types, functional annotations, Go ontology, MP, HPO, and exhibit pathway data includes in MolSig DB, LD Hub, lots of other data resources. And it's relatively straightforward for us to add data resources, and we've worked with Gene Network and others to bring in certain selections out of their databases as well. GeneWeaver supports 9 species. I'm probably going to see 10 of them if I count too closely, because it's true. Most of these are commonly used in the biomedical research area. So human, mouse, rat have the vast majority of the data, but we also have rhesus monkeys, Drosophila, C. elegans, and some other more agriculturally relevant species as well. We organize sets of tiers. This gives you a variety of quality, veracity of the data. And we're working on annotating each gene set with quantitative measures of expected number of true positives, false positives in each data record, and also the extent to which the whole genome was eligible to be included in that data set. And we could go into that a bit more. But these are kind of the 5 tiers that we think of in our data that we have taken from a high quality curated public resource, data that was machine generated by processing a public resource. So for this group in genenetwork.org, if we run a correlation of a trait to gene expression in a brain tissue, if we do that in mass, process all that data and put it into the database, those are tier 2 data for us. That's a post-processing, our unique post-processing and filtering of data from someone else's database. So it's fully reproducible from another database, but it's not exactly what's represented in that database. Public curated data are gene sets, usually entered individually, obtained from the literature, entered kind of one at a time, and advanced to public. They've passed our curatorial review. And then tier 4 data are gene sets that have been advanced by users to the public. So they're being shared with you, so you can use them immediately, but they have not yet been passed through our curatorial review. So use at your own risk. And tier 5 are data that you have uploaded for your own private use. Any analyses that you run are going to rely on that set of data in addition to all the other tiers, but only users that you've designated as members of your group to which you've shared that set of data can access it in their analyses. So
Elissa, can I point out something real quick here? Is one of the benefits of GeneWeaver, I think, is you could take your own gene set and compare it against a backdrop of many, many other studies. If you notice from this slide, we have lots and lots of private gene sets. We don't use those internally to do any kind of analysis. We don't really look at them. But as that's kind of a good steward, it's really great when we see people take their private gene sets and promote them to public. I think because everybody can share in that knowledge. So we always try to encourage that from our users.
Yes, especially if a group has done the hard work of curating a literature. We would love to see those data shared. Some of these, of course, are large numbers of gene sets emerging from analyses like gene co-expression analysis clusters and things. But yeah, we would love to see this number be closer to 0, and these public curated gene set numbers to look a lot more like this one. And that's up to our users to share those data and make them public for everyone else. This is the basic anatomy of a gene set. We've provided multiple levels of meta content for each gene set. Your text search for gene sets is going to interrogate all of these, but some of the controlled and structured annotations can be used analytically. So in this case, we have the tier designation and a gene set ID. We keep those gene set IDs stable. So if it's a data derived from a public source that's been updated, you'll have the same identifiers. The name of the gene set should be descriptive, something that you can wrap your head around. But we also provide a text-based description, and this we want to reflect the rules for getting a gene on this gene list. What does it mean to be a gene on this gene list? How did it get there experimentally? If there's thresholding involved or other statistical processing involved, we want that on there as well. We have publication information, and then we have annotation to a number of different ontologies beyond what you see here, so that you can choose controlled vocabulary terms that apply to your gene set. We indicate, now these are the genes on the set, we indicate the gene symbol. If you want to translate from one species to another, or to go from microarray identifiers that were originally used in the gene set, you can use the this page to actually translate all your identifiers to another identifier type. So it's a convenient feature here. Links to other gene set, to other external resources, and a number of different tools. Now, if you're having an emergency because our gene set is, say, misrepresenting the data, or has an issue with it, or there's problems with the description or title, or you want it to be reviewed, you can hit this request curation button and report that that issue exists. Yes.
Or Eric, can you show the URLs? So is there, for example, is there a URL that I can go directly to that anatomy of gene set?
Oh, this is of course a slide. I think
I'm going to paste a link to the page in the help documents that describe this.
I just want to point out that if you're writing a paper and you use GeneWeaver, you can cite it generically, which is great, obviously, and Eric and Elissa and team would hugely appreciate that, rather than just being totally buried somewhere. But I think the more active way to use GeneWeaver is to actually embed the URLs in your results and even your figure legends, so that if you're talking about a gene set, you now have a live link to that gene set in your figure legend. And it makes it much more interesting for the user, because they can immediately go to that gene set and maybe even annotate it if it's open to annotation.
Right, and so the GSID can be used to build the link to the actual gene set. So it is a nice way to provide a table of the data that were used in your analyses. So I think I mentioned there's a lot of ways of sharing and exchanging gene sets and data. This sort of schematic represents the relationships among people using GeneWeaver. So a user has access to all the curated gene sets and anything that they've individually uploaded, any of their own private gene sets, and anything that's been shared with them. Other users have only access to the gene sets that were provided to them by this user. So it's a way that you can expose only what you want to your colleagues, and that your analyses are going to see anything that your colleagues have shared with you in addition to the public resources. It does mean different users of GeneWeaver may get different results depending on who they're sharing data with. So it's good to have a lot of friends, I think, is the upshot. We talked about our generalized bipartite model. This is a representation of that from our original review in 2009, early in GeneWeaver history. But basically the idea is we represent the biological concepts as one partite set, and genes, gene products, and biological, like molecular components, in the other partite set. And then the algorithms apply to those. I think I elaborated on these functions earlier. This is what the tools actually look like. So GeneWeaver provides you a nice set of D3 generated graphics that are quite interactive and dynamic. Erich Baker and his students made a lot of these. And they allow you to visualize a lot of different outputs from the comparison of these gene sets. This is a hierarchical similarity graph. allows you to look at all the intersections among your sets, which species are underlying those intersections, whether it's one or more species. Jacquard overlap uses the Jacquard similarity statistic to pairwise compare all the gene sets that you've selected. There's tools for network-based clustering. There's MSETs written by Michael Saul when he was a trainee prior to joining my lab, but we've embedded that tool into GeneWeaver. This allows you to do something which you can't do with gene ontology, which is to test the hypothesis that a specific gene set is overrepresented among your results. So if I want to know, are genes involved in, well, dendritic branching overrepresented in my gene set? I can search for the gene ontology annotated gene set for dendritic branching if it exists. I can find the genes associated with that. Axonogenesis I know exists. And then I can say, okay, I've got my differentially expressed gene list. Does it contain more axonogenesis or dendritic branching genes than expected? And that's based on the universe of genes in the database, the annotations, and the probability of a gene set like yours, based on its size, containing that number of genes from another gene set. So it's a nice statistical test of a hypothesis in an annotated gene set.
Hey, Elissa?
Yes.
Can I ask about that last thing? So there must be some null distribution if it gives you a p-value, right?
That's correct.
And is that just assuming all genes are equally likely to be in a gene set, or do you try to take into account other kinds of things that would influence that, like, you know, gene size in some cases might be important, or whether or not a gene is represented on certain arrays that are sources of data, or, you know, there are a variety of things that could make it non-random which genes would appear in a gene set.
So in the Jacquard similarity overlap, for example, and so your question may be specific to MSET, but I'm going to take it on in a general way. In the Jacquard tool, we do use the appropriate reference gene set for estimating the comparison. Jacquard similarity is the positive match over the possible matches, right? And so if I'm comparing a zebrafish data set to a mouse data set, there's a lot of genes that don't have an ortholog.
Sure.
And so the set of possible matches is a lot smaller than the union of those two species genes sets from their entire genomes. So we do take that up pairwise in the Jacquard overlap. It's one of the things that makes this computationally quite intensive when we do these analyses. MSET, I'm going to have to defer to Erich or possibly to the paper itself. MSET does do resampling. It comes up with empirical null distributions for the gene set, given the genes in the gene set and the genes in the database. What it doesn't do is what you were suggesting, which is look at kind of sequence-based properties of genes, such as the size of the coding region. This tool set primarily is built around the abstraction of gene entity to gene function, and not necessarily digging into sequence-based comparison in many ways, because certain aspects of sequence are non-conserved, especially in our case where our interest in non-coding variation. Those variants are not conserved. So we're asking about the roles of genes in processes. And so, yes, there are sources of bias based on gene length. We would hope that those things are taken up at the level of putting those genes on the list, right? And scoring those genes and the magnitude of their association with the concept that's represented in the gene list. And then we can go and compare what we hope, and you know, it's just hope, what we hope are unbiased gene sets.
Right.
Right.
So I think you would agree with this statement then. A p-value that comes from some of these tests may or may not accurately represent the null distribution Isn't that right? Because if the input data sets didn't effectively control for these confounding factors, and there might be a lot of different confounding factors, right? We could sit here and play a game of thinking of all the sources of confound. Then the test that you're doing, because it assumes that those are accounted for in the data sets, wouldn't detect the problem and might give, for instance, a very optimistic p-value.
Well, only some of these tools actually give you p-values, and those tools do.
Yeah, no, that was what triggered me to ask those. It sounded
Yeah.
like you were assigning p-values, right?
Where possible, where a statistical analysis is run. We have looked at things like frequency of annotation. You know, so there was a time when you used another famous pathway resource where MYC would come up in all of your analyses, MYC, the transcription factor, and it's because it's connected to everything, and it is thus the glue that unites everything in every data set. And you're like, wow, how exciting, my gene set's connected to everything else through this one regulatory feature. We have looked at weighting by representation and things of that nature. I think the idea here is every species, every experiment, every platform has its own source of bias. And by aggregating highly heterogeneous data, we're getting around some of that bias because we're asking about the convergence of evidence. That bias would have to exist on every single gene set. So if I was comparing just microarray data, then they're all subject to uniform bias. But if I'm taking microarray data and functional annotation from mutation screens and genetic mapping results from multiple subpopulations, then I've freed myself from a lot of the biases that meta-analyses tend to fall into.
All right, I didn't mean to knock you off track, but when you brought up p-values, it seems to me that assigning p-values is complicated.
It is, and that's why we only do it where we're running statistical analyses. I mean, you know, there is a statistical probability of overlap, but like any set of genes, we're not going to go in and say, you know, what's the probability that gene sets from Abe Palmer's lab are real, you know, and go in and sort of reassign a p-value based on biases that we know are intrinsic to your data, right? So like that to me is out of scope. But again, the tiers, the data quality scores, the bias scores, proportion of true positives on each gene list, these are all ways that we can improve upon just kind of naively analyzing the sets that are in there. And some of those tools are in production.
Cool. Okay, good. Go on.
Okay.
Sorry.
I think I was elaborating on different tools and how pretty some of these graphics are. We have Boolean tools to combine, collapse, compare gene sets. So if you have a very large query, you entered your favorite word and you retrieve 500 gene sets, and you want just the highly connected genes that are present on a large proportion of those 500 gene sets, you can use the Boolean tool to compress them all into a single gene set. In my case, I've taken from Gene Network, where many different tissues or many different iterations of similar gene expression analyses were run. And I just want the consensus of those. And I could choose, do I want the union of all genes for which expression in the hippocampus on any one of Rob's data sets is correlated to my behavior? So I don't have to get into the details of which set do I choose. I can take either the union or the convergence of those gene sets. I mentioned that GeneWeaver has an API tool set, and that's what it is. So if you're using it from the web browser, if you're using it in a programmatic way, you're using the same set of tools, you're calling them on the same server. GeneWeaver has very detailed help and documentation. It's basically a nice manual. If you wanted to print it out in PDF, you can get a thick book to read at night. But I just wanted to refer you to this, and these slides will be available. But there is detailed documentation for using API and every other feature of GeneWeaver. This is pretty thoroughly written documentation. So if you go to this help link on the website, it's the missing manual for GeneWeaver, starting right from the beginning, and it's pretty interactive. Okay, I've preempted myself and summarized some of these tools. The way that the GeneWeaver works is you perform your search, you select gene sets, you'll add them to a project, and then you run one of these tools. All of these tools have options, and you can access them by expanding next to the tool itself. I'm going to present now a few examples that have come from using GeneWeaver. I think some of these are from various publications. So this is one from Jason Bouvier, where we looked at the intersection of alcohol preference and alcohol withdrawal at the suggestion of John Crabbe to say, are there any genes that are at the crossroads of these two related functions? And when we compare the outcomes of genetic analyses from multiple populations of mice, we were able to find regions that overlapped, predicting alcohol preference and withdrawal. And we just chose the first one, but there are actually several like this. There wasn't a lot of genomic data on alcohol withdrawal, but there were a lot of mutant screens on alcohol withdrawal seizures and other seizure-related phenomena. So we brought those data in, and there was gene expression data on alcohol preference. Anyway, if we look at this is this hierarchical biclic enumerator HiSim graph. When we look at the intersection of all of those gene sets, only one gene remains, and that's this adapter protein, AP3M2. You can see the gene sets that support its association. They come from mice, rat, and human. And when we knocked out that gene, or obtained a knockout that was made from a lab in Japan, we were able to see that there's actually a difference in alcohol preference and withdrawal between the knockout and controls. And we've since gone on to see that this is regulated by a variant in an enhancer. And because we have multiple mouse genetic populations in this analysis, we actually can refine them and say, is there any segregating variant that can account for this effect across multiple populations? And indeed there is. And although there isn't an enhancer variant in the human, there is a promoter variant. So you can actually, again, as I said, traverse this variant graph to find the variants in one species that might act like variants in another species, even though there's very poor conservation of the regulatory feature. This is another one. This is a refinement of a QTL mapped by Jill Rekla. And this is mapped in diversity outbred mice. No surprise to anyone who studied pain before, but TRPA1, a well-known pain gene, is found differentially expressed in the dorsal root ganglion, associated with sensory function in mutant mice, and accounts for the genetics underlying the QTL. This is another one that was recently published where we looked at multiple behavioral QTLs underlying addiction-related behaviors. We looked at differentially expressed genes in humans, and we found that RAB3B, vesicle-associated protein, is involved both in habituation and locomotor activation in response to cocaine. And again, this one was borne out experimentally in the lab. This is a very simple application of the GeneSet graph. Search for multiple genes. Just represent that bipartite graph. And you could see these are aging studies now, and they're aging across species. We're looking for genes that might be associated with longevity. And through many different contexts, one of those genes was identified, tested in worms, and extends the lifespan in worms. Talked about the hierarchical intersection graph. This is a very large example of that. Many different individual gene sets related to alcohol response and alcohol preference. And when we start looking at the intersections of those gene sets, we can see genes that are present on a large number of those gene sets. And again, testing those genes, we see abnormal alcohol response. And this is PAFAH1B1. lissencephaly gene. But one of the things that we did when we did this analysis is we said, okay, there's convergent evidence. This is came from some of our early work. There's convergent evidence for a lot of genes in the genome. They're always coming up on microarray experiments. They're always coming up in these mutant screens. But then the genes that are well known to be involved in alcohol response, annotated either through OMIM or mutant screens, are actually fewer in number, and they were mostly the favorite genes of people who deleted them and did prospective experiments. So we asked the question, of the genes that are highly are shared among many different experiments, which of them are previously known to be associated to alcohol response? And so the way this graph is set up, these are genes on individual gene lists. And on the second level of this are genes that are in the intersection of two lists. And as we go up, we're talking about genes on 3, 4, 5, and above. So the more highly connected a gene is, the more gene sets it's represented on, the higher up in this graph it's represented. So this node represents multiple gene sets containing genes that are connected to all of those sets. And in this case, the number of sets is 6. So what we see is the well-known previously annotated genes are on the gene lists that are discovered using expression data, but they're not the most frequently observed genes. If we start running these genomic experiments that are freed from bias in gene selection, we actually see that there's lots of convergent evidence for genes that are not previously known to be associated with the behavioral function. So that's a very broad kind of overview of some of the basic functions of GeneWeaver. I just want to point you to these slides that are going to be made available. And this is a detailed tutorial for you to use after the webinar to run through an exercise that's going to tell you where to click and what to do. You'll sign in and create a user account, and you see that it's got fairly thorough explanations of how to run some of these analyses using collections of gene sets that we've assembled for you. But we invite you then to extrapolate the exercise to a research question of your own interest and to use that to try to learn something new using these tools. And we're happy to assist people. You say, I've got a question, I'm trying to prioritize all the genetic variants I found over 5 years or something. We recently worked with Marissa Erringer and others to do this with some legacy data combined with some new sequencing data that they've generated to prioritize some anxiety-related genes. And we're very happy to interact with people to help them perform logical analyses of their work. So this is the tutorial, and again, I invite you to explore that on your own after the webinar. I'm gonna let's see, I'm gonna get out of the PowerPoint presentation now.
So Elissa, I'm gonna add one thing about the tutorial, and as people explore that, we spent a lot of time thinking about the questions of what needs to be computed versus what is computable. Of course, this is really theoretically unbounded space. So you can conceive of asking a question that is tremendously large. And the bandwidth we have for answering most of those questions we can do in real time. But don't be surprised if you ask a really large question that you'll have to come back to GeneWeaver tomorrow or the next day to get the answer and your results. So start out with small questions first, and then kind of gradually build up and get a sense for how long some of the analyses take.
Right. That's a very.
If somebody asks a really large question that's going to take a long time, do they have any indication of that?
They should. In theory, the tools will respond that the question exceeds the parameters and come back, tomorrow. Some questions, though, because of the way the tools scale, a couple of the tools are and log n type of scaling, so we can run them in. So if you have a very specific question that you come to us with that's really large, yeah, we have heavy iron. We can put our code on and give that answer that way.
Yeah, so some of those are more one-off collaborations and could emerge into some other development. But for your basic excessively large query, you'll run your analysis and go to this view results pane. And you can see it's been a couple months here for me, but you could see that I generated a graph on 6 gene sets a couple months ago.
If you're trying to share your screen, we actually are not seeing your screen.
Oh, right. Thank you, Rob. I have to press a few buttons again. Let's see. There we go. All right. So you should be looking now at GeneWeaver. And if I'll go back to the home page just so you can easily see what I did. I went to the Analyze Gene Sets pull down menu, and you could see my results. These are the results of analyses that I've run. And it's because it's been less than a few days since I've run it, I'll have to re-execute this. This is a fact, so I'll try to do it online. I can name it. You can see exactly, we can reproduce this exact result. This is also helpful if there's issues. If I'm not interested in this anymore, I could delete it, which cleans thing up. But with one click, I'm re-executing it. This is the same place that you would go to find the results of something that took too long to do. So if you feed us too much, come back tomorrow, go to view results. I don't know if we do a message for the completion of an analysis. It's fairly straightforward.
No, we do not.
There is in-app messaging, mostly around curation.
Could you just blow it up a little bit, or the max? We can see it, but we can't resolve the text at this scale.
Oh, okay. All right. Which text are you trying to read?
It's that
all,
Zoom thing where it kind of blurs images.
Yeah, that's good. That whatever mag you're at now is great.
Okay.
You're an iPad mag now.
I know it's blindingly large. Okay, so it's the second item in this analyze gene sets pull down. So a lot of times, say we're interested in this is where I get in trouble. I'm going to start doing analyses wildly while we talk. I've searched for cocaine. There's a very large number of cocaine-related gene sets, and I could see a gene list that I might be interested in. And I'll select that gene list. I'm going to just zoom out a little bit just so that we can see some of the basic buttons and functions.
Still legible.
Yes, I can. So I can find gene sets that are similar to this one quite rapidly, but I'll let it go. It's going to run.
It'll take a minute.
But if I've run a search, and I've selected now my 3 favorite gene sets from this, and you can take as many as you'd like, but in the interest of time, I now need to add these to a project. And I'll add them to either my existing project here named A cocaine, so it can get to the top of the list. But I might want to create a new project instead.
Called AAA cocaine.
Oh yeah, right. It should be AAA or AA Palmer cocaine.
Not as good.
And then I create that project. I'm not done yet. My project's been created. I have to go through a few things here. The appending things with A only works so often. In order to add these to that.
So Elissa is a great super user for us.
In order to add to AA Palmer cocaine, I now need to select the new project I just created and submit. If I then want to analyze those selections, I go to Analyze Gene Sets. And again, I have to find AA Palmer cocaine.
Got it.
Yep. And I can expand it. I now see those gene sets. I can chuck the ones I don't want. I can add more to it. And now I can access the tools. And again, these are some of our simple tools. I can expand them. I can work within a species or not. I can set some parameters on these tools. And again, the documentation is really detailed. So if you're looking at this, you don't know what these parameters mean, how they're applied, go back to that GeneWeaver help. So I set my favorite parameters or go with the defaults. I hit run.
You didn't
And
select
again,
any gene sets.
Huh?
You didn't select any gene sets.
See that? Yeah.
You have to,
The
yeah.
user error. These are 3 relatively small gene sets, which I expect may or may not come together. But we'll see on this gene set graph. So we're now just drawing the bipartite graph. And I see that there are some genes that are only on this gene list, but I have a large number of genes that are on the pair of gene lists. Now, the more gene lists I add, the more I'll find a small number of genes connected to a large number of gene sets. So, you know, you go and you continue searching for cocaine. I'm not going to attempt to run that live, but those are the types of examples that you can work through using the demos. Are there any other questions? I didn't go through the curation tools. There's a separate set of demo slides for it. But basically I can have a standing PubMed query, addiction genetics. I can run that query repeatedly. You can see what it does on PubMed, so you could pre-test it before you make it part of your work. You can rerun it. And now I can work with these gene lists. I can assign them to my curators, who might be the undergraduates in my lab during the summer. I can review the way they've described them, and then I can make them part of the database. So there's a lot of functionality for group curation, again, towards Erich's point of making these data usable and computable for a broader community of investigators.
So Elissa, there are a few, at least one question in the chat. The first one's from me. I could hold off and we could wait until till the top of the hour, or but keep going if you want to keep going, because this is great.
I think a few more things that people might not know to look for. It's kind of trying to hit the deeper points. This is where you're going to control users and groups. The account settings panel. So these are groups that were created to work together on various projects or papers. Some of these are private, some of them are public, shared among other users.
This is also the page you go to set your API key. It should be.
Yep, at the bottom, you can generate a new API key. You can change your email address if you'd like. How you receive notifications, in-app notifications from GeneWeaver. And then when you upload text, when you're entering your own gene sets, which I should probably go to next, you can choose what annotators are used to scan that text to recommend to you controlled vocabulary terms that we should use to annotate your gene set. So, or you yourself can choose those terms. So you can type the free text or paste in the abstract for your work, but then use these text annotators to choose the terms, highlight selected terms, and pick them out of ontologies. So I'm going to go back to manage gene sets. I talked about curation, which is how you might get a large set of gene sets out of the literature that may be untapped. But me personally, I ran some analyses. I got my differentially expressed genes. I want to upload them and do something with them. I would go right here to upload one gene set, or if it was a bulk analysis from, say, all the output of my WGCNA, I might have a batch of gene lists. Upload gene set. I'm going to enter the descriptive meta content. I give it a name. I'm trying to be humane to myself and my colleagues by giving it a name that makes sense, so some kind of title. You saw that all the graphs I made are quite small. You want to give it a figure label that's compact. It could be some camel case representation of the name. Genes are assigned to gene lists by many different scores and different analyses, p-values, q-values, correlation statistics. The effect size is the most diverse. Any real number can be used to associate a gene with a gene's, with a GeneSet ID. So if you haven't got a legitimate statistic, you can use this relative scaling provided by the effect size and put that in there. And then again, the description are basically, hopefully, somewhat reproducible description of how your genes got on your gene list. And then we can pull in all the publication details from a PubMed ID, so you can enter one in and retrieve that from PubMed directly if it's a published gene set, or you can go ahead and enter it all manually. Then this is where the magic happens. You can either keep your gene set private or keep it or make it public. I can share my gene set with my group. Choose one of the species, choose a gene identifier. We prefer that people upload the identifier that was actually used to put genes on your gene list. But in lieu of that, you can choose gene symbols, proteins, whatnot. Paste in or upload the 2-column file of your gene IDs and your scores. And then you go on to the review and approve. You'll see which identifiers matched or didn't match the database. You can make corrections.
So one thing I'd like to tell users too, we have a batch upload form where it has all the metadata and the gene sets as well. And if you're a heavy user, you want to submit lots of gene sets, the batch form is really convenient because now you can store what you've uploaded. So if there's an error in the process, you don't have to retype everything. So I would recommend, if you're a little computer savvy, to check this out.
Now we do store what you've uploaded. So
yes.
we redo those mappings. We will update those mappings. If you've used an ancient array and the mapping file got updated, you'll be upgraded to the new mappings. So again, this is the reason why we want to store the original identifier that was used, because when we update the mapping, that will get updated too. The thing that you know about, the truth is that probe matched that score, but the gene that was assigned to the probe may only do so for some number of years until that gene gets updated. So we want to kind of have the ability to future-proof what you've uploaded.
So Laura has a question about gene uploads on the chat. And so yeah, you can upload gene sets that have all sorts of values, and we can threshold those. So if half your gene sets have a value of 0, you don't want to include them, we can set a threshold for any number above what you prefer. So I prefer to upload all the genes and then threshold them at some point. I just use a threshold gene set.
And I don't remember if there was a token for threshold in the batch upload header.
No, they're set manually when they're uploaded.
I mean, are there advantages to uploading all of them in the long term if what you're doing is trying to make a public gene list that other people can take advantage of?
Yeah, I mean, they can copy your gene list and change the threshold, for example. You can subset a gene set and make a new threshold. So there are some advantages. The other thing is most of our tools were combinatorial. Once you go across species, the quantitative scores don't matter quite so much. There's some problems in trying to relate those quantitative scores. So we were really looking for convergence of categorical evidences. But it does make sense to kind of bring in the weights, and we did design the tool to support the use of those weights in an integrated way. So for example, to support a proper statistical meta-analysis from the effect sizes provided. To do that, we absolutely need all the scores for all the genes.
Yeah, Elissa, so my question is at the top of the chat if you want to look at it. But it basically has to do with isoforms of mRNAs and proteins. So what you described today was mainly gene level. And I think that that's the appropriate level right now. But I hope over the next several years, we'll really have a much deeper understanding of mRNA and protein isoforms. Laura is certainly working hard at that. We've all talked about it for more than a decade, but actually doing it has been problematic with short read technologies and RNA prep protocols. But I'm wondering if, for example, for BDNF and for this CERT, transcert, and a couple of other genes, they're well-known isoforms. And I know some are conserved and some aren't. And do you have a feel for where we'll be, or who's developing homology maps for isoforms? I don't know of any work on that, of that type.
It's a good question. It might be a question for the Alliance of Genome Resources, and it's certainly worth considering. And we are using the Alliance database for doing our orthology mapping. The challenge becomes the more granular the annotation of genes becomes, the less aggregation you get. So unless you see isoform data emerging in large numbers, and not just enumeration of isoforms, but implication of isoforms in disease-related traits, you don't actually get the ability to take advantage of kind of global integration, right? So the abstraction weakens the nature and detail of the relationship on the one hand, but on the other hand, it allows you to bring more data together. And we used to use the, I believe this came from Archimedes, but I may be wrong. Quote the, the fox knows many things, the hedgehog one big thing. So you kind of, the bipartite graph lumps together a large number of data sets in somewhat of a simplistic way on the one hand, but on the other hand, a very powerful way, because we're able to ask the question over all of these details: how often is a BDNF related gene involved in a trait? Now, if it's one subtype of the BDNF transcript, one isoform, because we're mostly relying on positive match in the combinatorial algorithms, the mismatch information kind of splits off in kind of like a partitioning sense, right? So you basically see aggregation when BDNF variants are implicated, and it kind of falls away when they're not. And so what you're able to do then is ask, well, when does it appear and when doesn't it appear? Because we're separating genes and gene sets simultaneously. So you could say, here are the conditions in which BDNF is represented. Here are the conditions in which BDNF is not represented. And if any of them have that commonality of having one of the two isoforms in the underlying data, then you know that there's an isoform related difference in the function.
Yeah, cool. Thanks. Good answer.
So you mentioned the Alliance for Genome, forget what the last term
Resources.
is.
Yeah.
Resources. So in their homology, do I remember right, they use like 11 different criteria or something like that?
Yes, they use 11 different, 10 or 11 different tools to map orthologs.
So are you swaying on the more conservative end or the more liberal end as far as if it's one of the 11, if it's all 11?
We've always been the more liberal type. Well, I mean, because our goal has always been to aggregate data. The disappointment in sort of, these gene expression results are sparse, right? And we're trying to look for convergence. So, and we're basically saying, if it's noise it shouldn't appear consistently. Now, there are some instances in which noise becomes consistent. So we've always been a little looser towards bringing data together rather than trying to get extraordinarily fastidious and have a high rate of false negatives.
Elissa, have you and team begun to integrate single cell RNA-seq specific gene sets semi-automatically?
We've discussed it with some groups, but we haven't done it. You know, for us, the value, and I'm gonna get into broken record mode, I suppose. Where there's a lot of data, the tools become quite powerful. Now, taking one single cell genes data set, you know, for like brain, and then using that to interpret the convergence of gene sets related to a disease or a behavioral condition is actually quite useful, right? Because then you're actually saying, okay, I've got differentially expressed genes in my bulk RNA-seq experiment. Now I want to annotate them with cell type. And, you know, I could do that with structure by cross comparing my gene list to the Allen Brain Atlas. But again, a cell type atlas would give me the ability to resolve it to the cellular resolution. So it's certainly something that we want to do, and data sets are emerging that will allow us to do that. And we haven't yet formally integrated any of them, but we certainly could.
Thanks. I'm just seeing if there are other questions. So we still have a lot of people online. So 38. So please.
Yeah, jump in. And I do want to say, we do have a focus group. You can run through the tutorial exercise, and we will be debriefing with the focus group in the next week or so. And so there'll be a scheduled session for that. So if you want to join that focus group now that you've heard the presentation and learned a little more about GeneWeaver, do respond to Laura Saba, and she'll add you into that group.
Yeah, I think this will be great. I highly recommend this. I hesitate to highly recommend it because I don't think we can handle more than 10 people, but there's a world of difference between hearing a presentation on a web service and actually becoming facile at using it. And it makes all the difference in terms of adding GeneWeaver functions and results into papers. I can't recommend it enough. And I know this from firsthand experience because I've heard Elissa give this talk about, I don't know, 5-10 times. And I still am a little bit of a klutz when it comes to using the cool features in GeneWeaver.
So I guess I'll ask a question if nobody from the audience has one. I'm always curious with these tools. You talked a little bit about the single cell RNA, but what else are the kind of the big picture goals or the big additions that you feel like should be added into this data? Are they tools, or are they more data sets, or what's your vision?
It's actually a bit of all three. It's three major things. I know you only asked me about two of them. You know, the first, of course, is more data, and more data. The most exceptional, you know, we have the GeneSet type, you know, data. So more diseases or more breadth on the diseases. So this community has generated a wealth of addiction genomics data across multiple species. And we want to show that those converge. We definitely want to evaluate the convergence of those, not just because we want to say, hey, we're right, but what we want to say is we can leverage the neurobiology of model organisms to interpret kind of global causal variants from human genetics. Where, when, what, how in the brain are these genes influencing to influence addiction-related behavior? So we definitely want more data of the type that you've seen today. The other thing we are doing, though, is again adding quite a bit of gene regulatory information and developing more functionality in the back end that allows us to directly relate variants. So bringing genetics to integrative functional genomics, and not just at the level of here's a positional window, it's got a lot of genes in it that were implicated by genetics, but here are variants that are associated with traits. That's going to, over time, give us a lot more genetic information. On the one hand, it's going to give us contextual information, and the data in GeneWeaver, again, become the backdrop to do all the experiments and all the phenotyping that you don't get to do in a human genetic study. So you could say, here are human genetic variants that are associated with addiction. And then we can drill down into GeneWeaver and say, here are the brain regions, the cell types, the specific context, and the mouse variants or rat variants that you might want to perturb that have the same effect. So now you can do experimental tests to understand mechanistically how that human genetic variant acts. And I mean, it sounds kind of like, look, I found a human genetic variant. Maybe I'll delete the gene. Maybe I'll put the variant in a cell. You don't really know what experiment to do. I'll say I don't know what experiment to do, but I could go into GeneWeaver, as I showed in several published examples, and say, okay, what I want to do is perturb this gene and see if the mice drink more alcohol, or see if they have a heightened hypothermia in response to alcohol. GeneWeaver gene sets tell me what experiment to do on the gene I didn't know I was interested in until yesterday morning. So now it's like I can go in there and say, okay, well, what is this gene involved in, and get really in-depth information about the context in which that's appeared. I think that was two things. The third thing is moving beyond the bipartite graph. And this is where we get into heterogeneous networks. And so there's a bioRxiv preprint where we've started to dig into this, which is to construct a heterogeneous graph of the relationships among genes, gene functions, diseases, variants, and traverse that heterogeneous graph. And so that's the third thing. That gives us a lot more flexibility in saying, here are genes that exist in the context of these functions. Here are genes that exist in the context of other genes. So more kind of guilt by association, if you will, but algorithms that allow us to not simply cut up the world into genes and gene sets. I promised three things. Here's a fourth, which is a set of prototypical tools that we've written to compare graphs. So we've done a lot of list and set comparisons, but we've also got network comparisons. So you have a gene co-expression network derived from, say, one population of inbred mice. We've actually compared across all of the recombinant inbred panels of mice brain gene expression to say which of these co-expression networks are consistently observed. If you look at co-expression networks derived from multiple experiments, or you want to take a co-expression network and compare it to a biological process network, you want to ask the question, which edges, which gene-gene relations are observed across experiments? And that's just a re-representation of the bipartite graph. Instead of genes and lists, we now have edges and networks. And we're applying the same algorithms to say which edges are frequently co-occurring on which graphs. And so that's a whole suite of prototypical tools that essentially flips the model to focus on those gene interactions. So those are our two priorities. There's 4 of them.
Thank you, Elissa.
Sure.
Any other questions from the audience?
Still have 25 on. Siamak, Sean, let me just pick on people I know. Jared, Hal, David.
Not logging off.
I mean, I'll throw my name in and ask a question if it's all right.
Sure.
So thanks, Elissa. I've always learned something when I hear your presentation. I'm a little embarrassed to say I had never heard of the Alliance of Genome Resources, so it's great to hear that come online. So the question really is, how are you starting to think about non-coding genes rather than non-coding variants with respect to using them in GeneWeaver? Like more circular RNAs are coming out now and they're being shown to be heavily conserved across species. But it's really hard to relate that sometimes across species particularly, but also within the context of existing or new genes as they're coming out and being identified. Like Laura and Boris had a nice long non-coding called LRAP. And if we wanted to identify how LRAP's important across species, but it's not there yet.
Well, this kind of, again, especially in the case of microRNAs, so let's talk about microRNAs. They're known to regulate transcripts, but they're not easily mapped to those transcripts, right? But this is a case where we have a set of genomic entities that can be mapped to its target. And that target's a functional gene product. And it's the functional gene product and its association to the disease that we're really interested in, but we find it through its connection to, say, a microRNA or other non-coding feature, right? So, for us, you could store all non-coding features as entities and say, okay, how many times do I see this non-coding RNA, these pseudogenes, et cetera? But they're more interesting in some ways as causal nodes that have target. And this is where a bipartite graph can be constraining, whereas a heterogeneous graph allows us to look at those relationships more naively. The answer is, there's a couple of answers. Technically, the bipartite algorithm can be applied. Say you had 1,000 or even 10 gene sets involving particular non-coding structures. And you can ask, where do they co-occur? If you upload a gene that's not recognized by the GeneWeaver database, you still get an identifier for it. And Eric's going to have to walk me back or elaborate on the process.
I think walk you back's probably better. Yes.
Okay.
No, but we can't assign identifiers to pretty much any entity. But the request is through me and not through the website.
Right. So, but you can then start comparing sets containing that identifier. The idea is, if it's not frequently evaluated experimentally, there's going to be nothing to match it.
And that was the point I was going to make, Elissa, is that the power comes from sharing things across species and gene sets. So as we add very bespoke identifiers, if there's no way to make a connection with another gene or object in the database, then it sits on an island by itself and it has little utility.
Right. And so identifying the targets of those structures actually connects them to the mainstream of the data in the database.
But we are happy to add objects at any time. You can send me a list and we can walk through that. We've added them, transcripts in the past, new species as well. If there are species that need to be added, as long as we can connect them to another species in the database, we're okay.
So to follow up on Sean's question, circRNAs and other more exotic, or I like the word bespoke, but bespoke variants. If you go into a database and have a good assay for a circular RNA, say in liver or brain, and you compute the correlations to all of 100,000 isoforms, you'll have a beautiful list. So then you can indirectly annotate it to all the other lists that are already in GeneWeaver. So it doesn't have to stay bespoke very long as it's got a real connection that's not a batch effect artifact, which 9 out of 10 times it'll be a batch effect artifact, but if it's not, you may be good to go.
I guess I was kind of thinking along the lines when I saw Elissa's slide for incorporating genetic variants, say using high C data, looking at known TAD regions. Could it be constrained by what TAD things are located in, for example? Or maybe even when you upload a list, you can identify genes that are species specific, and you might not see that in that hierarchical graph when you're trying to look towards the top. And you only you find it's only expressed in humans, and that would be good information for users to know. I found a really cool species specific that only applies within my model organism, and therefore that's why it doesn't overlap.
Right. And so some of that hierarchical similarity gives you that information, right?
Oh, great.
So you'll have a sub branch that's, say, only zebrafish. And that's good to know. And then you'll have places where zebrafish have good relationships that are convergent with what you see in human. So you can see where and when you do match, and you can ask the questions about why you might not match. We've often thought about this problem in the context of behavioral assays purporting to study the same behavioral construct. Using the genomic data, we can actually ask the question, say of many different assays purporting to assess anxiety-related behaviors, how many of them give us what type of evidence? Is that evidence similar? Are there particular data sets, laboratories, techniques that give us completely disparate evidence? And I didn't show these examples, but I think they may be represented in one of our preprints, Eric, where you can actually ask the question, then take a human mapping study or a human postmortem genomic study, and Spencer Huggett's recently done this with cocaine, and say, okay, let's see how the human transcriptome data aligns with the model organism data. And we could ask the question, which processes, so in major depressive disorder, we ask the question, what mouse behaviors give us genomic results that resemble differential expression in people with major depression disorder? And you can actually look at that overlap, and we see that chronic unpredictable mild stress paradigms, as you might expect, resemble human depression, but things that we've found using tail suspension tests or other kind of frowned upon assays don't actually resemble the depression-related behavior as much, or rather depression-related gene expression. So this gives us a way of saying not just, does the model organism assay look like the human assay? It tells us which assay to use. And so it might actually help us identify those assays that are appropriate rather than just discarding things on the basis of poor face validity.
Well, if there are no more questions, maybe we'll call this a wrap. Thank you very much, Elissa and Eric, for coming in and doing this presentation for us. It was really helpful. And we will have stuff up on the website so that you can link back to the video, or you can link to some of the materials.
Thank you very much. And again, if you want to join the focus group and work along with us, please get in touch with Laura.